Hello everybody, in this video I'm going to speak about meiosis. In the previous video I spoke about mitosis and many of you asked me to speak about meiosis, so in this video I'm going to explain this process in details. Let's first of all have a quick comparison between meiosis and mitosis. In the previous video, I told you that in mitosis is a process of cell division in which I get two exactly identical cells from a somatic cell. So here it is. I have a cell of 46 chromosomes, and from this cell I get at the end two exactly identical cells, and they are also identical to the mother cell, and they have 46 chromosomes each, while in meiosis the case is different. It's also a process of cell division, but at the end I get four unique cells, non-identical, and they are not identical also to the mother cell. This meiosis happens in the germ cells, and it gives me at the end, as I told you, from one somatic cell which has 46 chromosomes, I get four non-identical cells with 23 chromosomes each. How does it happen and where does it happen? First of all, you have to know that meiosis doesn't happen in the whole body. It happens only in germ cells inside the gonads. And it happens to give me the gametes, which are the sperm in male and ovum in female. This is the cell cycle of the meiosis of these cells, and as I told you, it happens in testes in male and in ovary in females, and it gives me at the end the sperm in male and the ovum in female. So how does it happen? Let's take, as we saw in the mitosis, let's take the cell cycle first. And here I want to tell you something. So if you didn't watch the mitosis video, I highly recommend you to go and watch the mitotic video, the mitosis video before, because it's really similar to the meiosis with a little small difference. So it's easier to understand the mitosis, and then you can understand the meiosis really easier. Here is the cell cycle. In the mitosis we had one mitotic phase and then the cytokinesis. The interphase is exactly the same in both mitosis and meiosis. The only difference here is that we have two phases of cell division instead of one phase in the mitosis. So let's go into details. In the interphase, there are no much events, so the cell is growing up basically, and there is also the duplication of the DNA material, which happens one time in both meiosis and mitosis. So this is the interphase. The interphase consists of three subphases, which are the G1 phase, the S phase, and the G2 phase. In the G1 phase, which is the first gap phase, from its name it's a gap phase, so nothing very important is happening in this phase. The cell is just growing up in volume, and the cell is metabolically active and it forms proteins and organelles. So as you see here, the cell is continuously growing, the volume of the cytoplasm is getting bigger, and then we have more proteins and organelles in the cytoplasm. This is basically what's happening in the first gap phase. Then we move to the synthetic phase. In the synthetic phase, there are two things are being synthesized. From its name, it's a synthetic phase, so something is being synthesized. These two things are, first of all, the DNA. So the DNA is being copied. Every single chromosome is being copied to a two sister chromatids. So I get from every chromosome two sister chromatids, and then I get double the amount of nucleic acid I have. And the second thing which is being synthesized in the synthetic phase is the centrosome. The centrosome is a structure that exists normally in the cytoplasm, and in the synthetic phase this structure is being synthesized and copied, and this is very important in order to form a structure called the spindle. These two centrosomes are responsible to create something called the spindle. They put small structures together called tubulin in order to form microtubules, and these microtubules with these two centrosomes are going to form something called spindle, and the spindle is very essential in the division of in the cell division. I will show you this later, but you have to know what you have to know here is that this centrosome is being copied in the synthetic phase. The third interphase from the third subphase from the interphase is the second gap phase. As I told you in the first gap phase, nothing important is happening. So the cell is getting bigger. The volume of the cytoplasm is getting bigger, and also there is the forming of more proteins and organelles. This is the second gap phase. In this point, we are at the end of the interphase. The interphase is exactly identical between the meiosis and the mitosis, so nothing changes between meiosis and mitosis in the interphase. At the end of the interphase, I get a big cell with 2 sets of DNA and 2 centrosomes. This is what I'm speaking about. So this is the end of the interphase. I have a big cell. I have 46 chromosomes, but every one of these chromosomes is made of 2 sister chromatids. So basically, I have double the amount of the DNA. It's like this. So every chromosome is made of 2 sister chromatids. So this is the first chromosome, this is the first pair. I have 23 pairs of chromosomes. This is the first pair, second pair, third pair, and every pair is made up of 2 chromosomes, and every chromosome is made up of 2 sister chromatids. So the DNA material is doubled. This is the end of the interphase. Now we will start with the first meiotic phase. In the first meiotic phase, the cell will be divided into 2 cells. But what you have to know is that in the meiotic phase, the 46 chromosomes will be separated into 2 sets of 23 chromosomes. So, as I showed you before, the chromosomes, these 46 chromosomes are made up of 23 pairs, and these pairs will be divided into, every pair will be divided into 2 single chromosomes, and every chromosome is going to go into a different cell. The meiotic phase is made up of 5 subphases. The prophase, prometaphase, metaphase, anaphase, and telophase. We are going to see what happens in every phase. As I told you at the end, so what you have to understand is that the 46 chromosomes are made of 2 sets of chromosomes. Every set of chromosomes consists of 23 chromosomes coming either from the father, so the paternal chromosomes, or from the mother, the maternal chromosomes. And in the first meiotic phase, every pair of chromosomes is going to be divided into 2. Let's see how does it happen. First of all, in the prophase, we have several events. First of all, the DNA will get condensed. What does it mean? This is the DNA chain. The DNA chain coils around proteins called histones. Every 8 histones with the DNA chain around it is called the nucleosome. These nucleosomes are coils together, and then they form something called supercoils. These supercoils are really condensed. The DNA is getting condensed in order to be easier for the DNA or for the nucleic acid to be placed on the spindle. So these are the 2 chromosomes. This is one pair of the chromosomes. This is the maternal and this is the paternal one. In the prophase, these 2 chromosomes pair together. So first of all, we have the DNA condensation, let's say, and then we have chromosomes pairing together, like this. So every 2 chromosomes will be paired together. So we have the chromosomes condensed together, and then every 2 homologous chromosome pair, and then the meiotic spindle forms. As I told you in the interphase, we have these 2 structures, which are the centrosomes. These are going to form these microtubules in order to produce something called the spindle. This spindle is essential in the cell division because, so here we have the spindle, and here we have the condensed DNA material, and every two chromosomes are pairing together. And then every pair of chromosome is going to be placed on the spindle. But this is going to happen in the prophase. So let's see what happens in the prophase. First of all, in order to get the DNA material free, because the DNA material is still inside the nucleus, so the nuclear envelope needs to degenerate. So first of all, we have the degeneration of the nuclear envelope, and then or the disintegration of the nuclear envelope, and then the chromosomes will move to be placed on the spindle, and every pair of chromosomes will be binded to these microtubules on the spindle on something called the kinetochore. Then at the end of the pro-metaphase, the spindle will be formed, and then the chromosomes pairs will be placed on the spindle. In the metaphase, which is the third phase from the meiotic phase, these two centrosomes will start to apply pulling forces on the chromosomes which are placed on the spindle. These two centrosomes will apply exactly equal pulling forces from both sides, and these pulling forces will so these forces will cause the chromosomes to be aligned in the center or in the middle of the spindle, and this is what we call the meiotic plate, because it's similar to a plate, and it happens because both centrosomes are applying equal forces, pulling forces on the chromosomes. Then because of these, so the centrosomes, the centrosome pulling forces, and then the metaphase the metaphase plate forms in the middle. And then because of these pulling forces in the anaphase, every pair of chromosomes will start to be separated. So the pairs of chromosomes will start to be separated. We have the separation of two homologous chromosomes. So every homologous chromosome will be pulled in one pole of the spindle, like this. And then at the end of the anaphase, we will get one centrosome and one set of DNA on every pole or every side of this cell. And in the telophase, there is the nuclear envelope, which is formed again on every set of DNA, and then the cell will start, so the cytoplasm and the cytoplasmic membrane of the cell will start to be divided. So this is the end of the meiotic phase. So basically in the meiotic phase, I had the separation of the two DNA sets. This is what is it. So in the prophase, pro-metaphase, there is the spindle which is formed, and then in the pro-metaphase, the chromosomes are placed on the spindle, and then in the metaphase, there are the pulling forces from both sides, and then in the anaphase, every pair of chromosome is being separated, and then in the telophase, the nuclear envelope forms again. This is simply the meiotic phase. This is meiosis. So meiosis is not really complicated. You just have to understand what's happening during it. Then at the end of the meiotic phase, we have the cytokinesis, and this is the first cytokinesis. And during the cytokinesis, the two cells are being simply separated from each other, and we will get two non-identical cells. As you see, they are non-identical with 23 chromosomes on each side. But what you have to know, and what's very important, that is that these 23 chromosomes on each side every chromosome has 2 sister chromatids. So as you see here, we have 23 chromosomes on cell number 1 and 23 chromosomes on cell number 2, and every chromosome is made up of 2 sister chromatids. At the end of the meiotic, the first meiotic phase and first cytokinesis, we will get this. At the beginning, we had one diploid cell. This is at the beginning of the cell cycle. We had one diploid cell with one pair of chromosomes. You can see here that at the beginning, the chromosomes were not duplicated. The duplication of the chromosomes happened in the interphase. And then at the end of the first cytokinesis, I got one chromosome of each pair, but this chromosome is made up of 2 sister chromatids. You have to understand the difference between these two, because there is a huge difference between these two images. Here I have one single chromosome, but I have pairs of chromosomes, one maternal and one paternal. And here I have only one chromosome from each pair, so I don't have pairs anymore, but every chromosome is made up of 2 sister chromatids. And this is at the end of the first cytokinesis. Now, the first meiotic phase is over. I got 2 cells of 23 chromosomes each, and now I move to the second meiotic phase. In the second meiotic phase, what happens is that every chromosome will be separated, so every 2 sister chromatids will be separated from each other. Let's see how does it the separation of 2 sister chromatids. Let's see how does it happen. This is the second meiotic phase. So in the beginning of the second meiotic phase, I have one single chromosome, but every chromosome is made up of 2 sister chromatids. And at the end of the second meiotic phase, I will get. So instead of getting one cell of 23 sister chromatids, I will have 23 single chromosomes on every side. The subphases of the second meiotic phase are exactly identical to the subphases in the first meiotic phase. They are exactly the same. What's happening is exactly the same. The chromosomes condensing again. But here I don't have the chromosomes pairing because I don't have pairs of chromosomes anymore. And then the meiotic spindle, the meiotic and not the mitotic. This is the meiotic spindle is forming. This is what happens in the prophase, is exactly the same as what happened in the first meiotic phase. The spindle is forming and the DNA material is being condensed. These are the tubulin subunits, as I showed you before. These tubulin subunits are being synthesized by the centrosomes. In the pro-metaphase, which is the second subphase from the second meiotic phase, the nuclear envelope is being disintegrated, and then the DNA material will go to be placed on the spindle. And the chromosomes will attach to the spindle in the kinetochore, like this. But here I won't have pairs of chromosomes anymore, but I will have single chromosomes made up of 2 sister chromatids, as you see here. In the metaphase, it's similar to what happened before. The 2 centrosomes will apply these pulling forces on both sides, making to form this metaphase plate. And then in the anaphase, these pulling forces are going to separate every 2 sister chromatids from each other. And like this. So every 2 sister chromatids will be separated from each other. And then in the telophase, the 2 cells will start to be, there is of course the nuclear envelope which is formed, and then the two cells will be separated from each other in the cytokinesis. The separation of two cells happens in the cytokinesis. At the end of the second cytokinesis, I will get two non-identical cells with one set of 23 chromosomes. Of course, here I have either Y or X, but I have one single chromosome from each pair, so I have 23 single chromosomes. In the meiosis, I start with a diploid zygote, a diploid cell. A diploid means that there are pairs of chromosomes, so every chromosome, so I have two sets of 23 chromosomes, one paternal and one maternal, and the chromosomes are paired. This is the diploid. This is what I had at the beginning of the cell cycle. Now I will summarize to you what happens. At the beginning of the interphase, I had a diploid cell. Then at the beginning of the meiotic phase, or at the end of the interphase, I got also diploid cell, but the chromosomes are doubled. So every chromosome is made up of two sister chromatids. After the first meiotic phase, the two chromosomes are separated from each other, so I don't have pairs anymore, but I have chromosomes made up of two sister chromatids. And then at the end of the second meiotic phase, I get 4 haploid cells. Haploid means that there is no pairs anymore, just single chromosomes. So the haploid cells, 4 haploid cells, they are non-identical, and every one of these have 23 chromosomes. Now, you might ask yourself why the 4 cells or the 4 haploid cells are non-identical. And this is very important. And now I'm going to explain to you the reason of this diversity or this variety of the cells at the end of the meiosis. The first reason why these cells are non-identical is because of the random separation of the paternal and the maternal chromosomes. As you see here, every pair is made up of one maternal and one paternal chromosomes. And these two sets are separated from each other. But, I mean, from every pair I get randomly, I choose, or there is randomly one of these two chromosomes going to one side and the other going to the other side. For example, I might get here the maternal for the first cell. I might get maternal, 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 paternal, paternal, maternal, paternal. You know, you understand what I mean? It's totally random separation of these two chromosomes from each other. So here the set of chromosome might be maternal or paternal. It's completely random. And to get this set, I will get 23 to the power 23, which is enormous, because on every chromosome, on every pair, I might get one of two. So the possibility at the end is 23 to the power 23 sets of DNA, and this is why the two cells coming out from the first meiotic phase are completely unique, and there are a lot of possibilities for these two cells. So as you see here, the second reason of this diversity or this variation is something called the crossing over. Crossing over happens in the first meiotic phase. So when these, as I told you, in the first meiotic phase, every two chromosomes pair together in order to be placed on the spindle. During this pairing, these two chromosomes exchange genetic materials. So at the end of this, I get two chromosomes with mixed genetic material. So I might get a chromosome with both maternal and paternal genetic material on it, and this is called crossing over. And I might get many crossing overs between two chromosomes. So at the second meiotic phase, when these chromosomes, so this chromosome is going to go to one cell and the other chromosome is going to go to the other cell. In the second meiotic phase, and when these two sister chromatids are going to be separated, I will also get 4 non-identical sister chromatids. So as you see, I have 4 non-identical sister chromatids, and this applies on the whole 23 pairs of chromosomes. And this is why this is the diploid cell at the beginning of the meiosis. At the beginning, this is the first meiotic phase. At the end of the first meiotic phase, I have 2 non-identical cells with 46 chromosomes, with 23 chromosomes each, and every chromosome is made up of 2 sister chromatids, and these sets are not identical. And then when these 2 sister chromatids are going to be separated, I will also get non-identical cells. And this is why from one diploid cell, I get 4 non-identical haploid cells, and these are the sperms and the ova. This is what happens in meiosis. This is everything I wanted to tell you about meiosis. I hope you enjoyed this video. If you really did, leave your comment and share this video with your friends and colleagues. And see you next time. Bye bye.